Welcome to our channel where biology meets clarity. In this video, we have selected 5 critical molecular technology questions from PIQ and explain in detail which we believe not only increase your marks in the exam but also concrete your core concept in this topic. So, without any further delay, let's get started. Then it about the codon optimization. So before going to know what is the codon optimization, we should have a claim. So why there is a need of codon optimization? So there before there is another term that is the codon biasness. So what is that biasness? So I guess most of you know about the degenerative nature of the codons. So there are 64 codons and they synthesize 20 amino acids. That means there must be more than one codon for it should be more than one for the amino acids. So like as an example, for the proline you can get 4 codons like E C U E E C E E E and E C G. So that all codon can end up by synthesizing the proline amino acids, but they'll have some biasness. Like for the human genome, if there is a mRNA, in most of the cases, when there is a need to synthesize the proline as the amino acid, so in that case, you will get more of the cases you will get CCA. In case all of that sequences can synthesize the proline. So in the when there is a need to synthesize the proline amino acid, so the all the codons will be same in the number. In most of the cases they should be in the same proportion. Like when there is a need to synthesize the proline, in 25% cases this will be there, this will be there, this and this. But in reality that doesn't happen. So here comes the codon biases. In most of the cases you will get the CCA codon. The 4, the CCU, CCC, CCA, CCG, anything could be there. But in most of the cases, when you want to synthesize the proline, you will get the CCN sequence. So that happens for the human, and in the least case, you will get the CCU, the CCU codon. So that is for the human. So for another organism, like for a bacteria or for a virus, the sequence could be changed. They might have a preference on the CCU and the least preference on the CCE. So while there is a need of that codon biasness, so that is like a defensive feature. When in the human system, the CCA get more preference. So the tRNA having the anticodon sequence of CCA would be higher in number. That would be higher in number. That would be in an abundant amount. But where for the TCU, the anticodon, the tRNA having the anticodon of CPU would be less in number. The number would be less. So the tRNA respective to the CCA would be higher and the respective to CPU would be lower in the case of human. But if this bacteria attacks you, so they would be needed this tRNA. So the anticodon that is having that tRNA that is having the anticodon of this sequence will get the more preference because the number of this codon is higher in the case of that bacteria. So if there is the tRNA having this anticodon sequence against this codon, if that is not present in a abundant number in that organism, they will not be able to translate in an efficient way. So the translation will be down. Whether the humans would be in a high. The human's genome, the translation of the human mRNA would be high, but in the case of bacteria that has come from the outside, so that anticodon having tRNA amount is lower. Because in most of the cases, the CCA codon is there to synthesize the proline for the bacteria. But the amount of tRNA to synthesize the proline is very much less because they are using this human as the host. So the efficient translation would not be there and the bacteria would not be able to make the infection. So that is like a kind of defensive system. So when there is a need of optimization, so there is the codon biasness and you want to remove this biasness. Why you need this? Like in the case of vaccine, you might have developed a vaccine against the corona, that is a mRNA vaccine, say mRNA vaccine, and it had the biasness like this. So the effective translation would not be there. You would not get the product of this mRNA. That might be the uh, outer core protein of the corona. You might have make a vaccine against the corona, that is a mRNA vaccine, and it can synthesize the outer core of the coronavirus, and you are trying to introduce the a defense system against the corona. But here, if the codon biasness is present like this, so the efficient translation would not be there, and you would not get that much amount of the outer protein, and your immunity would not be boosted. So here you need to maximize the protein yield in the heterologous form. Heterologous first, you are using the mRNA of a virus the corona, and you are introducing in the another host. And here you want the efficient transfer, efficient production of protein. Maximize the protein yield. So the option 1 would be the correct. So here is the chart of chromatographic technique, and you have to match here with the list 1 with the list 2. So let's start with the simplest one, that is the gel precursor. So most of you know about the gel precursor. So you want to segregate the protein will be there. And if I say about the STS page, that is a type of gel filtration. In that case, the proteins will be negatively charged due to the presence of STS. Now you will make a negative charge shell and will make a positive charge shell, and it will run on the another end, and thus it will be secreted, and which will be having the higher molecular mass. This will not be run too long. So having the high molecular mass, we'll make a band here, and the middle one will be there. The lower wall is gay. So which one is having the less mass, that will run more. So depending on the molecular size, interpretation differentiate the proteins. It is short and it is not that much heavy. That's why it can run the long way. So, and the next one is the diaffinity. What's the diaffinity? Diaffinity, you are using in an immobilized plate or a immobilized plate. You are using a dye. Dye, what? That is a specific kind of structures. Those will be immobilized. So, on the membrane, that is the immobilized membrane, and this kind of dyes are there, and they they those are there. So depending on the structure and the hydrophobicity, the the then the dye will be hydrophobic in nature. In some cases, if you want to separate the hydrophobic protein molecules or a specific kind, or the protein molecules that are having the specific structure, then you can use this diaffinity. So here you will run the proteins from that side, and the specific structure will come and bind in this. The and this, and the rest will go down, and thereafter it based on the affinity. So thereafter, changing the nature, like changing the pH or temperature, you can control the structure of your target protein, and thus then the affinity. If you are using the pH or applying the temperature, then the structure will be changed, and the structural affinity will not be there. So you can collect it. So it will go down at that case. So it depends on the structural hydrophobicity. So the next one is the, let's say, chromatofocusing. So in the chromatofocusing case, there will be the gradient of pH. Like this is the 1, let's say 2, 3, 3, 4, 5, like the 14. From 1 to 14 will be here. We all know about the charge and isoelectric points. So like if you are given protein, like if you have a protein and you are keeping it here on the one gradient portion. So here the pH is 1. In that place, in that box, the pH is 1, and you are keeping your protein here. So 1 is a too low pH, and in that pH your protein must be in a positively charged condition. You can do the reverse thing. You can keep your in here in the 14th, and in that case your protein will get a charge of negative. That is for the 3HH group, and this is for the NH3 group. So now if you apply the opposite charge on the another side, like for this case, if you apply the negative charge on this side and if it has a positive charge on this side, the protein will start running on that side. So when like for this protein, it is going towards the 14 period. So on the way, the pH gradient is there, like say for the 6, if this is the 6, and depending on the isoelectric point, and I guess you all know about this, the isoelectric point is that point when the pH of the protein reaches to 0. So there it is in positive condition, and the on the way the negative charge, like if it is in a positive 5 at that one position, so in the two position, the 2 pH position, make positive 4. So it will start losing it will start getting the electron, because it is going towards a isoelectric point. So the positive charge is getting lower and the negative charge is getting higher. So at a specific point, like if the 6 is the isoelectric point, then it will come till the 6 and it will stop here, because the net charge is 0. The net charge on it is 0. So it can't go toward the positive or negative. So depending on that, so in that chromatofocusing pair, so you are able to determine which one is the isolated point of your target protein. So the chromatofocusing is related with the charge and isolated point. Now the last one is the covalent bond. Sorry, the covalent bond, like a very basic covalent bond is present at the thiol group. Like here you have to have again a immobilized pattern, a immobilized paper or filter, whatever. So here you will take the particles which will have the free SH group, TSH group, that is the thiol group. And we all know that the protein, the amino acid on the cysteine has a free thiol group. It might not be free, but if the protein is in a very condensed form, it might not be free. It can form the disulfide bond inside the protein, but there will be chances for having a SH group in the protein. So if the SH group is free, so before applying to this the covalent chromatographic technique, we must have to treat the protein in such a way so that we can have a free SH group of our target protein. So that SH group will come here and form what? It will form a disulfide bond. So the bond will be like this. That is a strong covalent bond. And thus the thiol group, free thiol group can form the, and that is in a immobilized form. So it can attract, it can form the a strong covalent bond. So the thiol groups can form the strong covalent bond like the disulfide bond, and your target protein will remain in the matrix and the other proteins will go away. Thus, you can use the chromatographic technique depending on these different properties of protein. So depending on this, the one is correct. So here are certain statements regarding the PET technology, and you have to select which one is the incorrect one. So what is that PET technology? So it depends on the activity of fluorescent probe. Like one should be the donor and one should be the acceptor. How it is becoming donor and acceptor? So at first there should be the excitation light should come to the donor. So the excitation light should be with a specific wavelength. So that light should have some kind of energy that can transmit to the donor. Energy will be transmitted to the donor and the donor will be excited. So from the ground state, the donor will transfer into the excited state. And such a way, some heat energy will be produced and the rest they will emit. And when they will emit the rest, so the energy will be here. So the energy, so this light will have higher energy and this light will have lower energy. And this donor will come back to its normal state after emitting the light. So the wavelength of this light would be lower and the wavelength would be higher, because the duration between the wavelength and the energy of the light is in a inverse position. So this will produce the light and this light should be, so the wavelength will be higher there. So high wavelength and low energy light will be emitted from the donor, and this light will work as the excitation light for the acceptor. So the acceptor will take that light and it will be excited again. Like the same happened for the case of donor, and the acceptor will be excited and thereafter it will emit another light. So if the donor and acceptor are very close together, so in that case you will get this light. The detector, the light detector will be there to detect the light. It will get this light. But if those are not really connected or they are far away, then this light. So the emitted light from the donor, the detector will get, the detector will sense this light. So as an example, we can say like CAP and YAP. That is the cyan fluorescence bulb and it is the yellow fluorescence bulb. So here the excitation light that will come, its wavelength should be 434 nanometer. And this is a violet light. So after the excitation, it will emit a cyan light. So the cyan fluorescence bulb, and this cyan light will work as the excitation light for yellow fluorescence bulb. That will produce the yellow light. So if they are close together, then you will get the yellow light. If they are far away, then you will only get the cyan light, because we are only giving this light, this violet light. So as an example, so where you can use this effect technology as an example, if you want to see the binding between two proteins like the insulin, if it is going and getting bounded with its on receptor like the GPCR. So if the binding is happening, then you will get the yellow light because they are very close together. And there is the if the binding is not happening there or they are too far away, then you will not be able to get this yellow light. Instead, you will get this cyan light. So there is another term in a mathematical term. We can use this efficiency of the plate equals to the R0 by R0 plus R whole to the power 6. So here the R0 has a specific value. So that is like a constant and it depends on the pair. Like for this CFP and YFP pair, this R0's value would be 4.9 nanometer. It depends on the pair. For other pairs, that would be changed, but it will be constant. For specific pair, it would be only constant thing. So the efficiency is inversely proportional to 1 over R by 6. So the efficiency of this freight activity will get higher when they will come close together. So it is inversely proportional with the R to the power 6. So when the distance will be so less, then its efficiency will be too high. So now let's take the statements. So the first one is saying the energy of the excited molecule, that is the donor, passes directly to a nearby molecule, the acceptor. That is right. The energy transfer is possible only when donor and the acceptor are close to each other within 50 angstrom. So that is the R, the distance between two both donor and acceptor. The distance is within 50 angstrom, then they will be efficiently produce the freight activity. So that is also true. And here it is saying the petty inversely proportional to 6th power, and it is saying that directly proportional to the 6th power of the distance between donor and acceptor. So we have seen it is inversely proportional. So that is right. This one is wrong. So it would be the answer. Here is a question about the polymerase chain reaction, and which has the efficiency of 100%, and you have to determine the number of DNA amplicons after 25 cycles. So what is the basic difference between the PCR and that is the in situ replication to DNA replication? So in the in situ case, there will be all the enzymes like the DNA template would be there, and the DNA polymerase will be there, and the DMTPs will be there, NTPs will be there, and additionally, the one of the primer, both the primers should be there, and additionally, the helicase enzyme should also be there. That how it works? It is there to degrade the hydrogen bonds that is in between them. So the helicase will come and degrade the hydrogen bond, and the silver strand will come, and thereafter primers will go. That will be extended. The primers will be extended, and now after the successful round, there will be from a single DNA molecule, there will be 2 DNA molecules. So thus it happens in a semi-conservative model. But in the case of PCR, the important difference is there. You will use all the enzymes, the DNA polymerase and these DNTPs, and also the primer, but only the helicase will not be there. Instead, you will be using a machine that is called the thermal cycler. So there you give a specific heat that will come and melt the hydrogen bonds, and thus they will produce the single stranded DNA, and thereafter this primers will come and will run the replication. So if there is a single DNA molecule, so in a semi-conservative model, it will convert into 2 DNA molecules. In the next round, from each strand, there will be from the strands will be broken away and they will form another from each strand, there will be one more strand will be synthesized and the single strand will convert into a double stranded DNA. So this will happen like that. So each round you get the DNAs in a multiplication. So the same thing will happen here also. So if the cycle is going for the 25 times and the efficiency is 100%, so after 25 cycles, so this will go to in the 25 time. Now that you will write in this form, that 2 to the power 25, it will be to the power 25, not 100. It is a assertion and reason based question, and it is saying about the shuttle vector. So before going deep into it, we should have a basic knowledge on the vector. So what is the vector? Vector is a specific kind of DNA construction. So it is a technology to transfer a target DNA inside your target cell. Right? That is the specific vector construction. And your target gene should be included and you will transfer it to your target organism. So the organism is not able to express or produce this here your target protein, but it wants to have this from a target organism. That is an extra chromosomal part. Because without that is not the genetic material of your target organism. Without this, the organism can survive. So here, so that vector should have a specific kind of construction. I am not going much deep into it, but for a shuttle vector, that is a specific kind of construction that can go in both, like in eukaryotic and prokaryotic system. Like if we were designing a vector, it can go into the E. coli and yeast. So you are designing in such a way a vector is a construction of DNA. So the construction you are designing in such a way so that it can transfer both in the E. coli and in the yeast system. So if you want to say the expression, so got that construction of DNA should go through which process? The first one should be the replication. So if in your construction you are not giving any site for the replication start, if the replication that machineries those are responsible for producing or running the replication, if they are not able to target your DNA, if they are not able to access your DNA, or they are not able to get the specific site to start the replication, then the replication will not be started. And thereafter, no expression will be there. If the replication is not there, so the further processes like transcription, translation would not be present here. So the expression will be very much down regulated. From a theoretical copy, you will not be able to see the expression. So you should include both in that. That is the case for vector. But when you are designing a shuttle vector, so you will use in both the organism, like in E. coli and Lister's example. So you should include the origin of replication for both the organism. Like for E. coli, that is the eco. That for E. coli, the replication site is the origin of replication. The ODC should be there in the construction, and the ARS, autonomous replication site of the yeast should also be there. So both the sites should be there, so the replication machinery for E. coli can sense this site, and for the yeast they can sense this site. So both the sites should be included in the construction to make it a shuttle vector. if you are only giving the what is C, then you only can transform it into the equal. You will see the expression in equally but not in the east. And if you are doing the vice versa, only air is there, then you will see the expression only in the east, not in the equally. So to make it a shuttle vector, input both the origin of replication. So a shuttle vector can be used in two different host organisms. That is right. The shuttle vector contains only one origin of replication. That should not happen. That is wrong. So depending on this, the A is true or R is false. So that option is correct.